Hello everyone. My name is Rupesh Kesharwani, and today I will be talking about very interesting application of Oxford Nanopore. Last year we have published a paper about STR spy, our method, where we showcased that our method was able to detect 100% concordance or detect the STR in autosomes. And today we have some more features added in STR version 2 that we are going to see that. Um, so let's talk about some basics. So what are the STRs? So in our chromosome, there are some segments called STRs, which are basically DNA repeats, approximate 2 to 7 base pair long. It share approximate 3% of human genome, and from very long time, it considered as a gold standard in forensic investigation. Thanks to the repeat that every individuals are different. As you can see here is the example with the three individuals. STR1 has different number of repeats in a different individuals. As you can see, STR1 has 10, 14, and 5, and the same as the STR number 2, like 12, 7, and 10. So there are many applications involved in this STR technique and analysis. One of the example I can show you here, like in 9/11 case, that victim were identified using this STR technique. And this I just found on Google that a few months ago, two new victims were identified using this technique. Not only that, we always use this STR technique in forensic investigation, and one of the very famous case we found that in Golden State Killer, that this guy has committed crime in 1974 to 1986, so many murders and rape, and the case was resolved in 2018. All right, so there are many issues or challenges associated with this STR technique. These are listed here. So we already have talked about these challenges in our first paper. You can take a look. But briefly, like there could be a stutter or PCR artifact in your STR analysis, allele similarity or diversity. Sequence diversity, this is the main challenge that we have seen compared to the short read and the long read. That it could be like you can see some mutation or variability within the sequence. Loci could be the same, number of allele designation could be the same, but the sequence could be the different. And the current technique only consider the length-based allele designation and do not consider the sequence-based variability. Thanks to the nanopore and the long read, that we overcome with this problem. So we have utilized one of the smallest device from nanopore that's called Minan. So because it is portable and low startup cost, that you can proceed with this, and it has capability of on-site processing. So in forensic field, we often need to go on-site to collect the sample without contamination. And it also enables sequence-based variability that you cannot see in a short read. And thanks to the nanopore, that now the error rate is only 3%. But still there are some gap that there are no such forensic specific softwares available. That is the reason we developed our method STR Spy. So let's see about the method, how it works. So in STR version 1, we have used the niched reference sequences to build the database. And we put a lot of effort building these databases as this looks like in this example. The yellow highlighted sequence are STR sequence repeat, and we added some sequence around it, like upstream and downstream, in order to find the sequence variability. Because of the long read from nanopore, we are now able to find the variation. And the header of this is the locus, bracket repeat, and length-based allele designation. So once we obtain the read from Oxford Nanopore, we do the mapping against the human genome and the remap against our database that we have built. At the third step in STR Spy, we do some quantification and normalization, and we automatically way up detecting SNV using the X-Atlas tools. So X-Atlas tool we have developed in our laboratory, and it's an alternative way of GATK, and it's very fast. Please take a look. And once we obtain the normalized counts from this STR Spy, we do the genotyping and the ranking. So after the ranking, we also perform the benchmarking and the finding the STR effects if there are any. So this is the simplest result from STR Spy. As you can see, the green highlighted lines are the STRs, the true STRs, and the blue one is the possible stutter effect, but has the lower normalized values. That's the reason it is not the top ranked. So we opt the point 0.4 cutoff in STR-SPY that we have benchmarked using 15 PCR cycle and 30 PCR cycle. So at point 0.4 cutoff, we achieve the most optimal true alleles in our prediction. So where we left you last year, so this is the STR version 1 results. As you can see in this heat map, we achieved 100% concordance using the reference sample in 30 PCR cycles. So just to summarize that, this heat map, the blue boxes represent the true prediction from our method, that is homozygous or the heterozygous. False positive is incorrect prediction, or if you miss sometimes, so it's called false negative. So what we have the update in STR version 2. So I'm quite excited about it. So this is the large database up to now that we have integrated in our method. So from STR-seq bio projects, like all the 24 of autosome as well as the chromosome Y that was lacking in previous version. And we put a lot of effort doing this, and we have found so many cases like some issues and challenges we try to resolve here. So one of the main challenges was like nomenclature. So for example here, so in C truth set, we have found the length-based allele designation was 9 and 10, but in Oxford Nanopore, what we come up with the 9.1 and 10, and that is actually true, because this 9 became a historical event, and now it's 9.1. So I just came across this paper just recently published that talk about the problem and issues related in forensic science, investigation, that there are many challenges regarding the nomenclature. Not only that, another exciting feature in STR version 2 that we integrated chromosome Y. So chromosome Y is equally important as autosomes, that you can resolve the criminal cases such as like sexual assault. Not only that, but if you want to see the familial relationship among the male, then you can utilize this method as well using the chromosome Y. We also benchmark this, as you can see, as a chromosome, as an autosome, we also achieve the 100% concordance for the chromosome Y as well. Not only that, we also tried resolve using this method some complex STRs. As you can see, our method can resolve the biallelic loci and correctly predict it. As you can see in this example, this first and second, and not only that, this also resolved isoallele issues. That is in this example, like DYS391 second. If you see, there are three samples which share the same length-based allele designation, that is 31, but the sequence variability, because of the nanopore read, the long read, we are able to correctly predict those one. Okay, so not only that, we also try to see whether it is ready for prime time, that you can blindly believe on this tool and to go for the real sample issues. So we also have tested this STR method with case sample. So we have sequenced the swab, bone, and blood. So blood is actually only female samples. That's the reason the chromosome Y is lacking. So there are some F1 score you can see, like swab 98% for autosomes, and 87% for the chromosome Y. And the same as goes with the bone, 98 to 99%, and for the blood, 89%. Keep in mind that this is not the 100%, but I like to underline one point, that this is the benchmarking without correcting the database of historical event, as I showed you in previous slide. So we may increase the precision of predicting for the case sample as well. So we are going to talk more about in our upcoming paper. Okay, so just to conclude my talk, so using the nanopore and STR-SPY, we achieve 100% concordance, and our database include CODIS as well. So now you can investigate chromosome Y as well as autosome together. And we have enhanced our database, and very comprehensive database that we built using the STR-seq bio project. That includes the autosome of 24 and chromosome 26. And we also address the issue of isoallele, length-based typing techniques, sequence variation, stutter effect, and SNV detection. And this is freely available in our GitHub page and Docker image container if you want to use it. And I'd like to thank my team, Fritz, who guided me through all this bioinformatic analysis in building this pipeline. Our collaborator, Courtney Hall, Bhupe, who are the PhD student and put a lot of effort doing this wet lab part. Nicole and Ruxane for guiding through all the project. And I'd like to thank NIJ for the funding. Thank you.